Hello, welcome to the session on research and treatment development for ARSACS. First off, I would love to turn things over to my colleague Betsy Trainer, who's going to give us a welcome from the ARSACS Foundation.
Good afternoon, everyone. First and foremost, I want to thank the National Ataxia Foundation for hosting this webinar and giving ARSACS a platform today to share some information on research and treatment development for ARSACS. On behalf of the ARSACS Foundation, thank you for joining us today. My name is Betsy Trainer, and I sit on the board of directors of the ARSACS Foundation, which is based in Montreal, Canada. The foundation supports ARSACS research efforts all around the world in countries such as the UK, Portugal, Italy, and the US. You can read updates about the latest research efforts going on worldwide by visiting us at arsacs.com. And again, we have a patient registry there and serve patients, ARSACS patients around the world. So although our organization is based in Canada, it does serve ARSACS patients and support research efforts and grant funding all over the world. So thank you so much for joining us, and we look forward to hearing from Dr. Wolter today. Thanks, guys.
Thank you, Betsy. And hi, everyone. If we haven't had the opportunity to meet just yet, my name is Dr. Celeste Stewart. I'm the Patient Engagement Manager for the National Ataxia Foundation, and it's my privilege to be the moderator on this series where we get to learn more about different types of ataxia. And today, we're going to be learning more about ARSACS. It is my privilege to introduce our speaker today, Dr. Justin Wolter. He earned his PhD in Molecular and Cellular Biology in the lab of Marco Mangone at Arizona Sate University. The Mangone lab uses C. elegans as a model to understand how post-transcriptional gene regulation affects developmental processes. During this time, Justin learned to love basic science and gained a deep appreciation for the power of mortal organisms. With molecular tools in hand, Justin chose to expand his training in neuroscience at the University of North Carolina, Chapel Hill. Never able to settle on just one project, the rich environment of UNC allowed Justin to work with several labs simultaneously. He developed a CRISPR-based gene therapy for Angelman syndrome in the Zilka lab. He used mass spec to investigate molecular deficits in a childhood onset neurodegenerative ataxia in the UNC Catalyst for Rare Disease group. And he developed an approach to study how common genetic variations affect cellular phenotypes in the Stein lab. The results of these diverse experiences laid the groundwork for his projects in the Wolter Lab at the University of Wisconsin-Madison. And with that, Dr. Wolter, the floor is yours.
Thank you for the kind introduction. It is very much my honor to be here today, and hearing that people are coming from around the world really is one of the unique treasures about what I get to do each day, which is interact with people from around the world with different cultures, perspectives, and talk about something that is all very near and dear to my heart and important to all of us, which is biology. So today, the goals for today, what I wanted to communicate with you generally is what we know about the basic biology of ARSACS. So my approach to science and to studying ARSACS is really based on basic biological principles. And it's really my opinion that we can learn the most about how ARSACS works and progresses from a standpoint of basic biology. And of course, that basic biology, we use that to inform treatment strategies and developing those. And so I wanted to also outline a lot of the work that has been done by many, many ARSACS researchers and where we are today for a treatment for ARSACS. My expertise is in basic biology, so I'm going to focus on that. But there's an excellent webinar that was posted just last week by Nicolas Dupré and Elise Duchesne that talked much more about the clinical information. So I really encourage you to talk or to watch that, because they talk a lot about the things that I will not speak to today. So in thinking about where to start for a talk like this, it's always a little bit tricky for me, but in this context, it's very easy, and that is with the foundation itself. I was a postdoc at the University of North Carolina studying basic neuroscience, and then one day, probably about 8 years ago, my boss came up to me and said, have you ever heard of something called ARSACS or Saxon? How would you like to work on it? And that came to be because Sonia, who's the founder of the foundation, reached out to him, cold called, and asked if he would be interested in supporting research about ARSACS. And that real simple reach out really, changed the course of my career. and has really made me focus primarily on ARSACS and, for the rest of my scientific career going forward. And so, we definitely want to start with the foundation and end with it because in the world of ARSACS, the foundation is really a unique gem and a gift that really keeps us all focused on a singular task. That singular task that we're focused on is finding a treatment for ARSACS. I encourage everybody, if you haven't already, to really engage closely with the foundation, either online or in person. There's lots of resources on the ARSACS website to learn more about ARSACS, learn all about the different things that are going on in the community. Okay, so in my own education for ARSACS, the first place to look is what causes it. And in the context of ARSACS, the causal gene was identified about 25 years ago this month now as part of the Human Genome Project. It was one of the first genes that was discovered as part of a disease through that group. And the title of that paper tells you a little bit about it, which is that it's a spastic ataxia common in northeastern Quebec, and it's caused by mutations in a new gene encoding an 11.5 kilobase ORF, or open reading frame. This is a very odd title for several reasons. The most relevant one maybe for our talk is the last part, which is the 11.5 kb size. What that refers to is the size of this protein. And so if you look at all of the human proteins, there's about probably 100,000 of them. And you look at just on their size, plotted based on the number of amino acid residues in them, you can see that most are around the 500 amino acid size. But Saxon is around 4,500. So it's about 10 times larger than most genes. And in most publications, when they talk about protein size, proteins of that size are not even discussed because there's only a very, very small handful of them. So this really kind of defining feature of Saxon as a gene has really defined the research field, in that it is very difficult to use standard molecular tools and standard cellular tools to understand such a large protein. But that being said, many researchers have been trying to tackle this very challenging protein for quite some time, and I'm excited to share a little bit about that progress. So the protein Saxon, as I said, it's relatively large. And one of the things that is kind of mysterious about it is in that large protein, there are many different types of domains or regions that seem to do very different things. So I'm just pointing to kind of a few of the known regions here that do all sorts of different things. They're known to regulate protein turnover, to interact with nucleic acids such as DNA and RNA, interact with protein folding machinery. It interacts with mitochondria, the energy sources of cells. It seems to do lots and lots of different things. And so because of that, the biology of Saxon is quite complex. Combined with its large size, it leaves us with quite a technical and biologically challenging task at hand. So I'll say that this publication identified the first genetic variant in Saxon. This is called the del-T variant. It's right in the middle of the protein here. And the functional consequence of this mutation is that the tail end of the protein is not produced. And so as a whole, the protein levels are lost. So it is inherited in an autosomal recessive pattern in that offspring of two carriers that are people who are unaffected, about 50% of them will be carriers themselves, a quarter of those children will be unaffected, not be carriers, and then a quarter will be affected. ARSACS has joined a constellation of many different autosomal recessive ataxias at this point. It was one of the very early ones to be described, but over the last 25 years, there's been over 100 new proteins that have some overlapping features with Saxon and ARSACS, but also some very distinct features of them. And really, this changing slope, this gene discovery process has really been accelerated by molecular techniques. And so this is genetic analysis where you can take the DNA from a person and try to identify genes that are associated with the specific disease of interest. And so a very standard approach to that is where you take a large number of patients that are diagnosed with ataxia in the clinic, but don't have any known cause for it. And you just do a widespread genetic analysis, looking for lots of different genes that are that may be associated with that, those features. This is one example from Europe that took about 800 people, and they found ARSACS variants in about 13% of them. This is relevant because it's the second most common, at least in this cohort, autosomal recessive ataxia that was identified. And this sort of approach has been used throughout the world now, so well beyond its origins in just Quebec. Basically in every continent of the world, in every country, ARSACS has been identified now. And so initially research in ARSACS was centered on Quebec and Canada. And now just in the last, 15, 20 years or so, with the discovery of really, really, really diverse Saxon variants located around the world, that clinical cohort has grown substantially, and with it, as has the research population. So combining all of this together, one last thing that I'll point out is that the dates of these publications are all within the last few years. And so DNA sequencing and DNA analysis technology is growing exponentially in its ability to identify variants. And so it is highly likely that the number of ARSACS patients will continue to accelerate, and that the patient community and the research community will continue to grow alongside. And I think that's a really important lesson that I want to communicate today, which is that the ARSACS research community has been growing massively over the last handful of years. But humans are not alone. So I had the privilege of getting to know Carrie Eckenstedt, who's a PI at the University of Purdue, because last year she is a clinical geneticist for dogs. She deals with mysterious diseases that affect specific pedigrees in a large number of species. And she was working on an autosomal recessive neuronal degeneration that looked a lot like a cerebellar ataxia. And so she described a pretty amazing set of features that are nearly identical to what is seen in humans. One of the things that's kind of unique about Pyrenees is that they are roughly the size of a human. Their spinal cord is exactly the same length. And so a lot of the things that she discussed in there is highly relevant to people. Eventually, through a kind of careful analysis of pedigrees and DNA sequencing, they were able to identify a 4 base pair deletion that looks a lot like the deletion that is found in Quebec, that can completely describe the pathology and the mutation in these dogs. And so all this to say that Saxon is not unique to humans, or ARSACS is not unique to humans. It naturally occurs in dogs and likely throughout the animal kingdom as well. And we just don't know about it yet. So if you put all this together, a really nice review by Nicolas Dupré earlier this year combined all of the mutations that we have identified so far in Saxon. And this is roughly what they look like. There are many different types of variants, insertions of elements, deletion of some. There are individual nucleotide changes, so not a loss of information or a gain of information, but a change of that, of a variety of different kinds. Now, this map is very, very unique in the world of clinical genetics. Many times, individual genes, when they're associated with a specific disease, will have variants that localize in specific hotspots, that is, regions of the gene that are functionally crucial for its function. But Saxon, it's not like that. It happens everywhere. It is likely that the incredibly large size of Saxon means that it is just a large footprint for random variants that emerge over time, as they do in every human. And so it's likely that Saxon's just large size contributes to its relatively high prevalence in the world of autosomal recessive ataxias. Now, one of the great mysteries in our field is, why is there not a specific pattern of localization of these variants? And a kind of intriguing piece of information came a few years ago from the lab of Francesca Malteca, when she took a bunch of cell lines from lots of different ARSACS patients with a variety of different flavors of variant, and just looked for the presence of the protein itself, the presence of Saxin. And so that's what we're showing here, is that in control cells, you can see the protein levels are high. And then in basically every cell line that they looked at, with a little bit of variance, the protein is completely lost. And so this really is our starting ground for understanding what the molecular pathology is in ARSACS, is that it is the loss of function of this protein that causes ARSACS. So ARSACS joins the world of autosomal recessive ataxias that have many things in common, but also several things that are different. This is kind of a nice review that talks about the overlapping and distinct constellation of symptoms in recessive ataxias. But the one thing that is common across most of them is obviously it's a movement disorder, and so that implies involvement of the cerebellum and spinal cord. And so this is an MRI scan of a patient with ARSACS, and you can see that the cerebellum, this small object in the back of the brain here, one half of it is a little bit smaller than the bottom half. And this is atrophy of the anterior cerebellum. And this is coupled with changes in the spinal cord. In recent years, we've seen increases in the thickness of the pons, changes in eye retinal thickness, and kind of changes throughout the central nervous system. And so Saxin likely affects many different systems in the body. However, the cerebellum, atrophy of the cerebellum is really one of the earliest things and most consistent things we see across all of the different Saxin variants. And so that's really been the focus of most of the research, is to try to understand the function of Saxin in the cerebellum. So what I'm showing here is a picture of a mouse cerebellum sliced through the side of it. And so we're looking at it kind of from a profile view. And you can see quite a beautiful structure of the cerebellum that really defines its function in the context of kind of neuronal activity, and also its function in regulating different regions of or different behaviors. So maybe the most conspicuous feature of this are these little red dots here, and these are called Purkinje neurons. Purkinje neurons are really central to all cerebellar ataxias in that they are the neuron type that is lost over time. So it's really thought that the progressive nature of cerebellar ataxias is driven, if not primarily, largely by the loss of Purkinje neurons over time. And you can kind of see a picture of that here in one specific lobule. This is in the anterior lobule of the cerebellum. You can see that in a normal mouse, you get a nice, beautiful kind of architecture of Purkinje neurons, somewhat like beads on a string. And in the ARSACS mouse, you have breaks to that string where there's individual Purkinje neurons that have died. But one of the very unique and fascinating things about ARSACS, and why I really believe that ARSACS is a great model and a great context to understand neurodegeneration more largely, is that not all Purkinje neurons are equally affected. So as we look at the cerebellum here, there's certain types of these neurons that survive. And it's in that survival that I think a lot of us believe lies the answer in trying to find a kind of disease-modifying treatment here. So this is known as selective resilience or selective degeneration. And so the question kind of becomes, what makes certain neurons survive while others die? Now, this is not a universal, this is yet not unique to ARSACS. Most neurodegenerative diseases, some neurons are spared and some survive. But really, by looking and trying to investigate what it is that makes some resilient versus susceptible, we can hope to understand the basic biology better in order to design therapies. And so the first clue to this came at the very earliest days. You can kind of see it in the MRI finding here that the anterior region of the cerebellum has shrunken more than the posterior region. So there's specific regions that are globally, like kind of a large scale, that are differentially affected. And this plays out in the ARSACS mouse also, where you have the loss of Purkinje neurons just in the anterior lobules and not in the posterior. But there's many ways to separate out these different types of neurons. The cerebellum has a very unique kind of cellular architecture to it. One of the features of that is kind of this stripy pattern of different kind of functional units of Purkinje neurons, that is, groups of neurons that function together. And those functional units can be distinguished by looking at different proteins, such as this one is called ALDO-C or zebrin. It's called zebrin because those functional domains form nice stripy patterns along the cerebellar cortex. And this is present in humans, mice, basically every animal with the cerebellum. This has been observed. And what's unique about this also is that you can separate out the neurons that are zebrin positive or ALDO-C positive from those that are negative, and they are not equally susceptible. So the zebrin negative cells will die, while the zebrin positives are really unaffected, or at least their numbers are unaffected. And so this is a major research question in the field, which is to say, what makes specific subtypes of these neurons susceptible versus resilient? And really, the ability to molecularly and anatomically dissect Purkinje neurons into different subtypes is really truly unique for ARSACS. And so it provides a very unique basic science research opportunity to, one, understand what happens in the context of ARSACS and possibly leverage that information across the autosomal recessive cerebellar ataxias and maybe beyond into other neurodegenerative diseases. Okay, so the main question driving our field really is, what is the causal mechanism underlying ARSACS? That is, what has gone wrong and how can we fix it? And so to do this, researchers take a lot of different approaches. Historically, most of the work in the first probably 10 years or so of the ARSACS, since the discovery of saxin as the causal gene, was using patient fibroblasts and cell lines. These are relatively easy to collect from people. And you can do really nice kind of molecular and cellular analyses, and I'll show some of that. About 10 years on, the first ARSACS mouse model was developed by the lab of Bernard Bray at McGill. Since then, there has been a second ARSACS mouse model with a different sort of variant in it. And in the last year or two, there's been the beginnings of several different mouse models that will allow us to more fine-tunedly genetically manipulate saxin expression and try to really understand at a more high-resolution level what it is that saxin is doing. And then lastly, there's lots of other model organisms you can use. Mice are not the end-all be-all of preclinical testing. And so the foundation has really has turned to these vertebrate models for the variety of strengths that they offer, and I'll touch on some of those later. One thing that I'm not going to have time to talk on today, but which is critically important, is that the ARSACS Foundation has also just started funding iPSC, that is induced pluripotent stem cell derived models of ARSACS. So these are cells that are taken from ARSACS patients. They can be reprogrammed into pluripotent stem cells that can then be turned into cells of many different areas of the body, if not most of them. And so you can use the same cell line. You can study cardiac tissue, you can study skin, bone. In the case of us, neuronal differentiation, and in the context of Dr. Becker's work, specifically looking at turning these pluripotent stem cells into neurons of the cerebellum. So this is a really exciting and important research opportunity. The real strength in this is that it is done in the context of the human genome. And that is something that you do not get in any of the model organisms. And it's something that really substantially influences the biology of ARSACS that cannot be modeled in other models, in other species. Okay, so with the goal of trying to describe what is the causal deficit in ARSACS, really right away from cell work, the first thing that people really observed was that cellular structure, that is the cytoskeleton, which provides the physical structure of cells, is largely disrupted in ARSACS. So normally this is a patient fibroblast cell line, just a skin cell. You can see a nice filamentous structure of the cytoskeleton. This keeps everything rigid, and it also provides tracks and the kind of foundation of all of the infrastructure to move the things around a cell that need to be moved around. And in the context of saxin cells, you see just somewhat of a collapse of that cytoskeletal structure. Somewhat intriguingly, that collapse is hollow on the inside. And so we've seen that there's different types of proteins that are trapped on the inside of this that might influence the ability for different molecular and cellular cargoes to move around the cell. It's not just this one specific cytoskeletal protein that we've seen, called here vimentin, but probably the one that's most relevant for ARSACS at the time or currently is neurofilament. This is the neuronal version of these intermediate filaments, and that also shows this sort of phenotype, this sort of kind of condensation within cells. So when we look at how the loss of Saxon could do this, one of the things that we see, Saxon is what I'm showing here in green, and kind of zooming in on one of these kind of end feet of the cell. You can see the nice filamentous structure of the cytoskeleton, and Saxon seems to decorate that cytoskeleton. So while we don't know precisely what its role is in regulating the cytoskeleton, it seems like it provides a structural component in addition to likely other protein regulation functions. And so really it seems to be holding the structure of the cell together and intact. It's distributed evenly across cells. And in the absence of that kind of scaffolding, the cellular architecture kind of collapses in on itself. Now, this is just in cell lines. What do we see when we turn to the brain? And so again, I'm showing the cerebellum of the ARSACS mouse here, looking at that neurofilament staining. And you can see, quite clearly in the context of ARSACS, that you see a similar accumulation and aggregation of that neurofilament cytoskeleton in Purkinje neurons, labeled like the beads on the string here, but also in lots of other neuronal cell types as well. So it is likely that ARSACS affects many, many, many different types of neurons, not just in the cerebellum, but pretty much everywhere that Saxon's expressed. And so the constellation of features that is seen in ARSACS can be attributed to lots of different neuronal systems that we really have very, very little knowledge of at this point. So the functional consequence of this, you could imagine that there might be lots of them. So one of them that my lab has studied a lot in collaboration with the Chapel Lab at Queen's College of London is looking at how things move throughout the cells. And so looking at live cell imaging, so this is a cell that is alive roughly in real time, you can see the movement of these things called microtubules. And microtubules are another class of cytoskeletal protein that are responsible for delivering things along those tracks. One of the things that they deliver along those tracks are mitochondria. And so if you've done any research on ARSACS on the internet, you'll probably see a lot of research about mitochondrial function. And it is certainly true that mitochondria are deeply entwined in ARSACS. What we see is that there is a large change in the movement of mitochondria in ARSACS neurons. And so what we're labeling here is specifically just the mitochondria themselves. So you can see them kind of moving around. In the context of a normal cell, they seem to move across these long processes in between the cell bodies from one cell to another. These are analogous to the axons and the synapses that you'd see in a neuron in the brain. And then in the context of a Saxon knockout cell, the movement of those mitochondria are affected. They're not as fast. There's not as many of them in the cell process. And so really, this is a deficit that can cause a lot of damage if the neuron does not have enough energy to do what it needs to do. So another consequence of that, which is quite relevant to what we see in the brain, is also other structural proteins that physically anchor the cell into its environment are also not in the right place and time. And so this is just a cell stuck on a two-dimensional surface in a dish. And this protein called vinculin here is localized to the regions of the cell where the cell sticks to things. Every cell needs to adhere to its extracellular environment. And this is a protein that really is responsible for sticking to things. And you can see in the context of a normal cell, wherever you see one of these red stripes, that's where the cell is physically stuck to the dish. And in the context of the ARSACS cells, you see the collapse of the cytoskeleton, and along with that, you get a loss of that localization pattern of these adhesion proteins into the surface of the plate. And so this is extremely relevant because in the context of the brain, the physical connection between a neuron and its extracellular space is how the brain and neurons functions, is that neurons need to send long projections out to different regions of the brain and to different neurons, and they need to physically contact those, or at least get extremely, extremely close to them so that proteins can interact across those cells and facilitate the flow of information across neurons. So what I'm showing here is right in the middle of the cerebellum of the ARSACS mouse. Everything in red here are the synapses. These are the long structures at the very end of a Purkinje neuron, and they're connecting onto another neuron right in the middle of the cerebellum. Now, these other neurons, they're called the deep cerebellar nucleus. They project everywhere else in the brain, in the central nervous system. So in the spine, the hippocampus, cortex, you name it. So really the interaction between Purkinje neurons and these neurons are really the central signaling hub for everything that the cerebellum does. And so any disruption to that process would affect the output of the cerebellum. And in fact, when we look at the ARSACS, the synapses in this region, you can see quite clearly that their shape and size are affected, as is their number. And this is long before Purkinje neurons start dying in the context of ARSACS. So it seems to be that one of the causal deficits from a cellular standpoint in ARSACS is a breakdown of neuronal connectivity. And this really shapes how we think about designing therapies and how we think about the ability to reverse or affect disease progression, is that this needs to be one of the primary targets. Okay, so along with the structure that we saw changes, we also saw changes in the localization of specific proteins that are responsible for sticking synapses between two cells together. And so one of them is just called integrin here. Normally in the Purkinje neurons, you can see that it's kind of loosely distributed throughout the dendritic arbor of these cells. But in the Saxon knockout, or in the ARSACS mouse, you see just a big accumulation of them, suggesting that this synaptic adhesion protein is not getting to the right place. And we can track the motion of these proteins throughout the different regions of Purkinje neurons and along the cerebellum as they move. And long story short, what we see is that there's a mislocalization of a lot of these proteins that are responsible for synapses to physically stick to each other. Okay, so that's a little bit about the cell biology of what my lab has been working on. But at the same time, there's many other labs that have been contributing to this work. And so over the last several decades, we have come to the realization that Saxon does lots of things. It interacts with lots of parts of the cell, and it interacts with lots of different types of proteins. That's just kind of summarized here. The functional consequence of that also seems to be far-reaching, in that basically every major aspect of cellular biology that we've looked at, I mean the collective field of ARSACS researchers, basically every type of cellular dysfunction that we've looked at, you can find it in ARSACS, which is to say you get a breakdown of mitochondrial localization, synaptic loss, deficits in protein turnover, changes in mitochondrial structure, the presence of oxidative stress, proteins being turned over incorrectly, et cetera, et cetera, et cetera. So, while we are able to nicely identify these things and characterize them in the context of cells, it really makes it quite challenging to understand what is the thing that is the most, that is causal. What is the thing that is actually at the heart of all of this? The other thing that's somewhat perplexing, not just about ARSACS, but in neurodegeneration as a whole, is that all of the things that we see in the context of ARSACS are also observed more or less in every other neurodegenerative disease. So, neurodegeneration is defined by neuronal death, but protein aggregation, just like in neurofilament, is common across most neurodegenerative diseases. Changes in synapses, aberrant protein turnover, cytoskeletal, mitochondria, et cetera, et cetera, et cetera. These are features that are observed in nearly all neurodegenerative diseases. And so this really begs the question that has shaped several of our research programs, which is to say, if everything has changed, what does that tell you about the basic biology? So how do we overcome the fact that everything seems to be affected? And from our perspective, one of the best ways to do that is to keep looking backwards in time. And that is to say, most patients come into the clinic after their symptoms have shown up, but in context of most neurodegenerative diseases, ARSACS included, that progression has unfolded over the course of years, if not decades. it's likely that there's something very, very early in the progression of the disease, long before you can see any deficits, that will tell you about the causal foundation for this disease. And so this is really the kind of central idea of my lab's research moving forward. I am not a clinician. I work with mice and cell lines, but to really kind of push the priorities of the foundation forward, we partnered with our clinical partners in Tübingen, Madison and David Mengel, who provided really carefully matched human patient samples that we could compare back to our mouse studies. And so we devised this strategy where we would march our way backwards in time and try to understand really what are the causal molecular deficits, what are the first thing that we can see in the context of the cerebellum that might point to therapeutic strategies that we can it can point to specific places where we could intervene from a therapeutic standpoint. So to do this, beyond cell biology, you might have heard this term omics, which is to say that omics is a kind of a broad stroke term to say, let's look at everything that we can see in a cell all at the same time. And so omics is a way to capture the molecular footprint and landscape of a cell using techniques that don't look at one gene or one protein, but look at it, all of them, or as many of them as you can, all at the same time. So these sort of approaches, you can apply them to DNA. And so this is genomics and DNA sequencing technologies that leads to the identification of a lot of the different variants. But you can also apply that to every level of molecular biology. So the genes that are transcribed and turned into RNA, you can measure the levels of genes going up and down in the context of ARSACS. You can also look at how that RNA is turned into proteins, which at the end of the day are the things that are driving most of the activities of cells. And so there's tools at basically every step along this way that give you just a huge set of data. And so we decided to take that really broad approach and look at it in the context of ARSACS over time. We wanted to do it, of course, in the brain, but perhaps some of the more clinically relevant pieces of data that you can get are ones that will be relevant to clinical trials and tracking disease progression and the effect of specific treatments. And so biofluids such as cerebral spinal fluid and blood can be readily collected from people and mice. Our strategy here was to have a deep understanding of what's happening in the brain, see if we can correlate that with changes in cerebral spinal fluid and plasma, both in mice and patients, and then correlate that back with clinical outcomes that we have from those same people to try to understand what is the molecular signature along the progression of the disease, that one, we can understand the basic biology, two, that we can hopefully identify specific regions or specific therapeutic targets, and then three, leverage that information to track the efficacy of therapies that are being developed. And fortunately, the ARSACS Foundation was interested in this idea and funded our team for the last few years for this project. There's a lot of stuff in here. I'm just going to focus on one of them that I think is really foundational for not just my lab, but what we're excited to share with the entire ARSACS community. And that's looking at really one data set that I think has been incredibly informative. So really one of the challenges of these omics technologies is that they usually rely on many, many thousands of different cells. And so you analyze lots of different cells all at the same time. Well, the problem comes when you have a complex structure like a brain. Really, there's many different cell types and there's lots of things going on at the same time. And so our strategy has really been pushed by emerging technologies, especially single cell RNA sequencing, that allow you to take a heterogeneous tissue like the brain, isolate individual cells out of that, and then measure the expression of thousands of genes in each cell. So this technology came out probably about 10 years ago now, but really has come to the forefront of neuroscience in the last 5 years, and really is reshaping a lot of what we know about basic biology, especially neurodegeneration. And so we did this in the context of ARSACS. It allows us to basically identify all the major brain cell types. Each dot here is an individual cell that we analyzed out of the ARSACS brain, and you can see many, many different cell types. And what this allows us to do is start to understand the kind of cause-effect relationships for some of the things that we see. So for example, if you just look at expression of the gene Saxin, it is very highly expressed in cells that we know are neurons, and it's most highly expressed in Purkinje neurons. And so this can really explain why Purkinje neurons are specifically susceptible, or at least explain a part of that. On the other hand, most of the other non-neuronal cell types, such as astrocytes and glia, don't really express a high level of Saxin. And so when we're trying to understand therapeutic strategies, there are many therapies that are targeted towards not neurons necessarily, but the neuroimmune system. Those therapies will not likely be directly acting on the causal molecular deficit, but more in the systemic response. So this is just kind of a simplified example of the sort of data that you can get out of this. Okay, so with a little bit more granular, just to give a sense of this, the punchline of this is that the brain is a very dynamic place, and ARSACS progression is also very dynamic. And so when we look at these a long time, both before the onset of motor deficits in mice, and then before the onset of cell loss, and ask what's changing over time, the very earliest stages, of course, you can see changes in the number of genes that are going up or down in the context of Purkinje neurons. So every dot is a different gene. You can see hundreds of genes that are affected at the very earliest stages of the disease, and other cell types a little bit, but not a ton. And then as time goes on, those dynamics shift markedly. So you see a change in the gene expression profile of Purkinje neurons. So this suggests that something is happening at this transition between neurons staying alive to dying, that we can try to understand to intervene at those specific time points. You can see how things like astrocytes which deliver nutrients to neurons, form the blood-brain barrier, do lots of crucial things in the brain, how those are also largely affected, even though they don't express high levels of Saxin. And then we can see really surprising things, such as the involvement of the vasculature. So the interaction between the brain, the blood flowing through the brain, and the cells that are contributing to that fluid flow, they are also largely affected. And so this is really kind of a surprising finding that I think is going to shape a lot of our understanding of basic biology. Okay, so that's kind of bringing everybody up to speed on what we know about the biology here. I'm sensitive of time, so I'm going to move a little bit quickly through the therapeutic, where we are from a therapeutic standpoint. But all this to say that the standard progression for drug development will be difficult in rare diseases, in that there are not many tens of thousands of people to go through clinical trials. We do not have the opportunity to screen tens of thousands of compounds in the context of ARSACS. So really our strategy and of the field as a whole is to take what we know from the basic biology, take what's already been discovered from other FDA-approved clinical trials and drugs, and really combine the two to identify really strongly motivated candidate drugs that can be used in ARSACS, and this is called drug repurposing. Okay, one of the areas where drug repurposing and drug screening has been incredibly helpful is in the context of zebrafish. Why zebrafish, you might ask? One, they have relatively complex nervous systems as we do. Their larvae are completely transparent, so you can really image and see the entire brain, not just from a slicing standpoint, but as a whole, and that has major advantages over mice. They share a lot of the pathological features of mammals. But what's really amazing about zebrafish is that you can test drugs very quickly. So you can assess their movement of these zebrafish when they're as little as 120 hours old. You can simply put the drug into the water and it goes right through the entire system of the fish. And some of the drugs that are of interest for the ARSACS community do seem to have an effect on movement behavior in the fish. Some major progress has come in the last 2 years in that the first disease-modifying treatments in the mouse models have been published. And these are from the labs of Francesca Malteca and Anne McKinney and Alana Watt. And they target seemingly different things. One of them, ceftriaxone here, is an antibiotic well known for its ability to affect glutamate uptake and calcium signaling. Francesca's lab tested post-symptomatically or delivered the drug post-symptomatically and looked at both movement, change in movement over time, and in the survival of Purkinje neurons, and reported successful results. This is an antibiotic, and so it's not appropriate for long-term treatment in humans, but it really points to some molecular pathways that may be clinically actionable. On the other side, the labs of Anne and Alana took a different approach, which was to test this compound called MitoQ. MitoQ is a dietary supplement, which is known to be neuroprotective in lots of different preclinical models. And similarly, if you give the mice MitoQ through water for many, many weeks, you can see a reversal, or at least a decrease in the progression of movement symptoms, coupled with cellular improvements in cellular pathology. One of the things that's interesting about MitoQ is you can get it on Amazon. So it is not necessarily a drug. It is a dietary supplement. And this is a really important thing to think about, not necessarily for MitoQ, in that its efficacy in humans has not firmly been established as it has in mice. But what's important about this is that there's lots of things that can presumably affect disease progression, which I will talk about briefly in a moment. Okay, the first FDA-approved drug for recessive ataxia came out within the last year or two. This is Skyclaris. So Skyclaris was shown in a reasonably sized small clinical trial of about 100 patients, that basically this drug slowed the onset of disease progression. It did not seem to reverse symptoms, but at least over the course of a year, certainly slowed progression. And now that patients at like the national or the International Congress of Ataxia Research and other ataxia conferences, we're starting to hear about Friedreich's ataxia patients that have now been taking this for some time and do report positive results in a subset of their symptoms. But it doesn't treat the underlying causal deficit of the disease. So without getting too much into the details here for time, I'll say that Skyclaris targets this molecular pathway called NRF2, which is in general an antioxidant or cellular stress response pathway. This has been really well established to be a pathway that's affected in Friedreich's ataxia, and Skyclaris directly interacts with the deficits in that molecular pathway. By stabilizing this protein called NRF2 and ensuring that it gets into the nucleus and turns on a whole bunch of different genes and related to antioxidant stress. So NRF2, the NRF2 gene is really only expressed not in neurons, but in astrocytes, microglia, the supporting cells of the brain. And so this can explain why it's influencing the systemic nature of this disease. And that should kind of inform our expectations about it in such studies. But nevertheless, we were able to identify that NRF2 signaling is also affected in ARSACS. And so this motivated us, or motivated the foundation to really reach out to a couple teams to test Skyclaris and then also other drugs that are known to target NRF2 signaling as a way to target inflammation in ARSACS. And these studies are underway now. But one thing that I really want to point out, which is kind of taking a page off of the MitoQ story, was that antioxidants and targeting inflammation can be done from many, many different angles. And inflammation seems to be associated with many, many different diseases and the progression of many different diseases, chief among them neurodegeneration. And so there's lots of healthy lifestyle choices that can be made that can help attenuate inflammation. This can be diet, exercise, et cetera, et cetera. There's all sorts of literature online. Now, this is really important in the context of ARSACS because I've heard it said, I don't know the exact motivation behind this, but about 50% of the disease progression, when it starts, how fast it progresses, about 50% of it can be defined by genetics. The other 50%, roughly 30–40%, can be defined by environmental factors, be it the rest of the genome, the specific, not just the specific variant that you have, but all the other constellation of variants that you have, your environment, and other choices that are made to help attenuate processes such as inflammation. And so, beyond specific, clinical treatments, there's lots of information out there that can help guide your personalized kind of strategy for how to address ARSACS. Okay, so our strategy for testing DMF really is what everybody in the field should do and is doing, is that when you test something in a mouse, it should mirror what you do in human trials. So DMF as a Nrf2 agonist, you have a step up in human patients for multiple sclerosis, and then we test in the long term. How we do this in mice is basically put them through a series of behavioral tests. So we watch how they move in spinning wheels, how fast they can stay on for, how strong they are. We can assess their ability to their speed to move and balance across beams, and then their strength, their ability to hold on upside down, given the ability to hang. And so, maybe some of these are familiar to you from clinical testing, but we try to take motor phenotypes that are relevant to patients and assess them in our mice as well. Okay, the last treatment that I want to talk about is really quite an exciting one, and one that I think touches on what is missing in a lot of the therapies that we have so far, which is really replacing what is lost. That is, a lot of the molecules that are tested are aimed at inflammation or the immune system, or things that are somewhat secondary to the primary insult in neurons. But what if we could turn on, replace the loss of saxin? And so there's a lot of progress in that, not just in ARSACS, but across the neuroscience field these days. And really the workhorse for this strategy is adeno-associated virus. This is a virus that we are all exposed to. It is not pathogenic. We have different antibodies against it in our system right now. It's somewhat commensal to a portion of the population. And it is relatively small. It's hollow inside, and so you can leverage some sort of genetic tools to engineer specific genes that you want to deliver inside that virus. And so you can imagine taking, instead of the virus genome that is normally there, replacing it with your gene of interest, and so saxin. So this is really the power of AAV, is that you can customize whatever genetic cargo you want, and there's many, many tools that can do this these days. So just in the last month or two, the very first FDA-approved AAV gene therapy for a central nervous system disorder, where the AAV was delivered directly to the brain, was just approved. And so this is incredibly exciting in that it's going to give a lot of momentum to this idea in the field. The other thing that's incredibly amazing about it is that there's a lot of question about rare diseases and how treatable they actually are. From a sheer patient number standpoint and a clinical trial standpoint. What stands out about this specific disease, about AADC, is that it is ultra rare. So rarer than ARSACS, rarer than most of the other recessive ataxias. In the literature, there's about 350 patients that are known. But nevertheless, this disease had a successful AAV gene replacement therapy that was FDA approved and now available for these patients. And so this is a great bit of momentum for this approach. However, there are some limitations to it that challenge its use in saxin, or in ARSACS, and that is that it's a relatively small virus. And so you can only package things that are about 1,200 amino acids in size. Saxin is many times larger than that. And so the question becomes, what regions of saxin do you absolutely have to replace that? Fortunately, the lab, Benoit Gentil and Heather Durham, have been working on this for many years now, which is taking small pieces of saxin and assessing which of them are sufficient to rescue some of the traits that we see in ARSAC cells. And over the last few years, there's been some progress where they're now testing these strategies in mouse models for ARSACS, where they've packaged just a chunk of saxin into an AAV and are delivering that. So this is really stands at the forefront of a truly disease-modifying therapy in that it targets the causal molecular deficit. Advantages, obvious, it causes the causal deficit. You can cater it to specific cells, so it's not like a drug that's going to have systemic-wide side effects. You can really cater it through genetic tools to the specific cells you care about. And somewhat remarkably, it's a single treatment. This AAV, once it's delivered to the brain, in mice and primates anyway, seems to last for the lifetime of the organism from a single treatment. And so it's really hopeful that you can just deliver AAV once and have it have a long-term effect. However, obviously, there's challenges with the size, packaging limits of it. It is extremely expensive, and so other AAV-based gene therapies are over $1 million a treatment. And then also it's a relatively new sort of delivery strategy. And so safety and delivery, while they've been demonstrated in some diseases, are not always guaranteed. And so for some other disorders, safety has been an issue. And so those are challenges that the field really needs to overcome. Okay. So I guess in summary of this kind of whirlwind information, really the main things that I want to communicate is that the ARSACS research community is incredibly strong and growing. Really, this is pushed 100% by the foundation. And so, really encourage everybody listening to engage and participate with the patient foundations, both the National Ataxia Foundation and the ARSACS Foundation, because it really keeps that momentum going. Really, from a scientific standpoint, we try to use the complex biology of ARSACS and different strategies to inform our development of specific therapies and the different strategies that we can take based on what we know about the biology. And then the last thing that I really want to communicate is that disease-altering therapies are inevitable. We are going to find something that alters the progression of this disease. While we don't have anything at this time, the momentum really is at our backs. The wind is at our backs, and it is only a matter of time before we find something that works for ARSACS. So, I guess I will end on that hopeful note. I will say that what can you do? How can you engage in this research? The ARSACS community, or the ARSACS Foundation website, has lots of links to different clinical trial or different kind of natural history studies, different ways to engage both the scientific and research community and fundraising communities, and then also what you can do personally, such as exercises and some sort of things that you can do at home to help take some agency over your progression of ARSACS. And then the last thing that I'll say, which is from a very personal standpoint, that I would strongly encourage is, talk to a scientist, reach out to us. I work in a lab at a university. I do not have the privileges of talking with, people that are affected with ARSACS very often. And honestly, it's something that is, I think, we suffer from. It's been the most rewarding and impactful times of my scientific career have been when I've sat down with patients and families in the few opportunities that I've had to do that, and really kind of hear and talk to them and learn from them. So I really strongly encourage you, as the kind of patient population, the patient community, engage us. And really don't hesitate to reach out, phone call, email, whatever. Myself and lots of the other ARSACS researchers, if not all of us, really benefit from you and talking to you. So please don't be strangers. And with that, I'd like to thank the broader community, the many labs that have contributed to the work I showed today, especially of course, the ARSACS research community. So Sonia and Betsy and all the researchers and people that are behind the scenes of this. It is a fantastically strong community. And so it's really on their backs that I make the statement that a therapy is inevitable. And yes, so thank you for your time.
Thank you so much, Dr. Wolter. I realize we're at time, but would you have some time for some rapid fire questions?
Absolutely, I will do my best.
So first off, just a question for NAF. There's someone who asked if ARSACS is the same as ARCA type 1, which that is a different disorder, but never fear. We have already had an information session on ARCA1, also known as SYNE1 ataxia. We'll be putting that information in the chat. So question for you, Dr. Wolter. Have there ever been a statistical analysis done about where the majority of people of ARSACS are in the world?
A statistical analysis, no. Kind of thorough genomic technologies have spread around the world. They're adopted first, of course, in Europe and other kind of financially and research-rich countries, but now they are becoming more affordable and are spreading around the world. So statistically, no, we don't really know what the prevalence is in every country, but anecdotally, it has been observed pretty much everywhere. Every time it's been looked for, it's been seen.
Good to know. And then the next question is, so if there's no protein, Saxon, regardless of the variant, why do symptoms and disease severity vary so much between patients? What's known about this?
Yeah, so in my opinion, is one of the most important questions, not just in ARSACS, but across nearly every human disease. So my lab studies neurodevelopment, including autism, and similar to autism, ARSACS has a huge range of progression. Even people with the exact same variant, the Delta variant from Quebec, there can be a huge difference in age of onset, the progression, et cetera, et cetera. This is true of every disease in humans. And so it's a foundational question that is important to all of us and really kind of the focus of most of my research. So one of the kind of most intuitive answer to that question, there's kind of two directions we can think of it. One is genetics and one is the environment. And so the genetic background, we think about genetic background in the context of the variant that you have in Saxon. Of course, different types of variants will influence that protein level more or less. We don't really know for sure, but it's kind of intuitive, and I think it's starting to be better understood that variants that remove maybe 95% of the protein are not as bad as the variants that affect 100% of the protein. And so even modulating levels of Saxon, even 5–10%, I think it will bear fruit to say that they will have slower disease progression based on that. Now, why that is, we really don't know why. And so patients, learning more about genetic variants and contributing to research, be it from donating cells and samples, et cetera, to help us understand how that progression and why that progression is different across people will be absolutely crucial. The second thing in genetics is not just the one variant you have, but the other 3 billion variants that you have. So your genome is incredibly complex. Between any two people, there's about 5 to 6 million common variants that make you who you are. Your height, weight, hair color, eye color, your ability to metabolize drugs, et cetera, et cetera. Those variants are not pathogenic. They contribute to basic biology, but they likely influence the outcomes of a rare, very highly penetrant variant like Saxon. So that sort of genetic approach is relatively hard to do in rare diseases. You need lots of people to participate. And so if there's one thing that I really look for the ARSACS community to embrace more, is not just genetic testing for the variant, but broader looking genomic analyses to look at common variants that can be assessed relatively cheaply, but then also whole genome sequencing that can really be leveraged to identify lots of different types of variants that might interact with your specific variant to define what pushes you one direction or the other. It's a great question.
Thanks for explaining that complexity to us. Our next question, have you ever received any human brain donations to study ARSACS?
So this is one of the challenges, and thank you so much for this question. This has been one of the limitations of ARSACS research, is that there are only a small number of postmortem samples available. Small number meaning single digits, if not more than 2 or 3. And so, they're in specific labs, and so it sometimes can be difficult to get them. And even if you have access to them or collaborate with those labs, as we have in the past, you're still talking about two people, and the underlying biology of humans is so complicated and so different just one human to another that really the sample sizes that we need to really explore this in the context of humans just don't exist. Um, I'll leave it there, but say that that is a huge limitation, that really only the patient community can rectify, I guess.
And if you're looking for more information on donating your brain to ataxia research, we have information available on our website on how to get in contact with researchers who could benefit from these samples. And then our next question, when do you expect clinical trials for people to begin, similar to treatments like Skyclaris or vatiquinone that are being tested in patients with other forms of ataxia?
Yeah, so while I am not directly involved in clinical trials in the patient side of things, it's expected or at least anticipated that clinical trials for a couple different things, or at least one thing, will presume or will start in the next year or two, is what I've been told. And so that involves big consortia such as from the National Ataxia Foundation, but also European agencies and Canadian and US. And so it's a very complicated landscape. On top of that, it's still uncertain what is going to be the lead compound, I think. That sort of information is still furiously being tested by many labs. And so while there are candidates that will likely emerge out of that in the next year or so, on the back end, the foundations and the clinical community is preparing for it. So one of the major emphasis and pushes for the ARSACS Foundation is clinical trial readiness. And for the last, at least 10 years, they've been really making sure that they have all of the information that they need in hand on the day that a disease-modifying drug is identified, so that they can push forward relatively rapidly. They are ready. The community is ready. It just kind of depends on the backs of testing drugs. So
100%. And we have one final question today. Will these clinical trials, and this is for you, Dr. Wolter, as well as you, Betsy, will these clinical trials, do you think, will they focus on adults, children, or a combination of both?
I can jump in there just from the perspective of the ARSACS Foundation. We are, in fact, embarking on our first human clinical trial, which will take place in Canada and have a cohort of adult Canadians only. And we're doing adults for obvious reasons, safety and efficacy. It's easier to start with a small group of adults, and then hopefully, if it's successful, follow on with a younger cohort. But as soon as we can publicly release that information to the ARSACS community, we will certainly update everybody. But we are up and running and fully intend on launching a human clinical trial, hopefully within the next year.
Well, we will stay tuned for more information. That's all the time we have for today, folks. So Dr. Wolter, Betsy, thank you so much for joining us today and teaching us more about ARSACS. We really appreciate it.
pleasure. Thank you.
Thanks, everyone.
Yes, and to everyone who was on this call, I hope you learned something valuable today or learned something a little bit more about ARSACS. Wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care.